I think that this would be a good time to talk about the near-infrared spectroscopy instrument and why it was designed, which we've talked about somewhat already with regard to the importance of lipid core plaque, both for vulnerable plaques in future events as well as for the adverse events in the cath lab. You've spent a good part of the past decade working on an instrument designed to detect the presence of lipid within a plaque, and now have given us this beautiful combined multimodality instrument that gives us both plaque composition in terms of lipid content as well as intravascular ultrasound. Tell us what the NIRS-IVUS instrument is, how it works, and what data it may provide that can help us both in terms of making decisions in the cath lab and performing PCI, as well as potentially identifying lesions in patients at risk for future events.
Okay. It required internet technology. It required the latest lasers, latest optical detectors, latest algorithms. And it's probably only possible now to do such a measurement in the human coronary. But as you know, it's FDA approved. I do recall 3 years ago, Jim, you said it's a beautiful instrument to measure lipid, but it has no structure, and you need to merge it with IVUS. And then we began a 3-year engineering task to combine it with IVUS, and today you saw the results, and I think we've got that part done now.
So the beauty of the instrument is that it's simple to use. And the design was really well thought out because it really recapitulates the kind of an instrument that we've been using as an IVUS catheter for the past 15 years. So it looks like an IVUS catheter, talks like, quacks like an IVUS catheter. So it's basically the same setup. You take it out of the package, you connect it to the pullback device, you plug it in, and you're ready to go. You put it over a monorail wire, and you do a pullback, and you get instantaneously on the console, you get a beautiful IVUS image, and the IVUS image now has evolved to be, in my opinion, equivalent to the best-in-class grayscale IVUS. It's important to recite what data there is to support that these signals that come out on the NIRS-IVUS instrument are established against histology truth to be lipid core plaque, because it's the only instrument that has been approved by the FDA for the detection of lipid core plaque. This is based on elegant histopathologic studies done against autopsy specimens where they cut diseased arteries stem to stern and proved that there was a lipid core plaque, and then validated the yellow signals that show up on the chemogram on the NIRS instruments to be highly predictive of a lipid core plaque established by histology truth. So
let me just give the background on that. You know, as you stated, the near-infrared was rigorously validated versus autopsy specimens. That took about 2 years to do, that study, in many hearts. And that led to the FDA very specifically stating this is approved for detection of lipid core plaque. And they actually stated that this was the first device that could do that. There are other devices approved for broader characterization of coronaries, of course, but the special part about near-infrared is it's specifically for lipid core.
So, as with most new instruments, one asks questions as to, well, how will this data be helpful? We had some very interesting observations, and this came from a patient taken care of by my partner, Simon Dixon at Beaumont. He had a patient with acute coronary syndrome who came to the cath lab, had a very large right coronary artery that had a complex lesion in its midpoint. And he treated it in the standard way, put a wire down, and he blew up the balloon to pre-dilate it. And before he did the balloon angioplasty, he did a NIRS study of it, and this plaque just lit up. It had a giant lipid core. And it was not surprising, so it was probably a vulnerable plaque. This patient had been asymptomatic and then suddenly had chest pain and came in with this acute coronary syndrome, so it validates this hypothesis in this patient of a vulnerable plaque that was not flow limiting, that then it explodes and forms this unstable plaque, and the patient presents with this acute MI. And so he went and performed this procedure and blew up the balloon in the right coronary artery, as one typically does before putting in the stent. And after the balloon was deflated, the patient crashed in the cath lab. The patient had abrupt, total no reflow in the right coronary artery. The patient became profoundly bradycardic, went into complete heart block. The blood pressure plummeted and was very ill in the cath lab for about 10 or 15 minutes. Through his expertise, Dr. Dixon was able to stabilize the patient, implanted a stent, got an excellent result. The patient did have a peri-procedural infarction, which we've talked about. It occurs in anywhere from 5 to 12 or 15% of patients. And so this was the first signal to us that, gosh, maybe these lipid core plaques are a potential problem in terms of these peri-procedural MIs. Well, then as you delve into the literature, it wasn't a new idea. There had been 20 papers written using intravascular ultrasound and other technologies to show that these yellow lipid-laden plaques were at the greatest risk for distal embolization. So this has led to what I think may be the first potential clinical application for pre-procedural plaque characterization to try to reduce this not uncommon problem of peri-procedural MI by identifying those plaques at highest risk, namely heavily lipid-laden plaques versus fibrous or less lipid-laden plaques, and potentially identifying lesions that might be protected with a distal protection device, a filter, to prevent that downstream embolization of this lipid content. And we now have some data to suggest that we can identify those lesions at highest risk by using NIRS-IVUS analysis.